All right, great to be here with you all. Thanks for staying here. I realized, and I think Dr. Lutz realizes also, that we are standing between you and a nice glass of wine or a glass of beer or whatnot, or maybe a dinner. So we're going to try to make it as exciting as possible. All right, let's move forward. So I wanted to bring to you today some perspectives from industry, and especially at the company that I work for, Eli Lilly, we have begun an initiative to look more at how can we change how we do drug development to perhaps be more efficient. So we're really we're looking outside the typical box of how we do drug development, or as someone suggested earlier today, throwing the box out altogether. So I've entitled this Influencing the Future Success of Medical and Drug Development Research. So I'm going to start out telling you a little bit about the why. What are some of the things that we are challenged with? And to begin with, timely valued medicines is an internal TVM is a word or a slogan that we use inside Lilly, talking about how we're going to get to our human patients. It's a patient-centric, data-driven quality approach. And I'll preface this with the fact that I'm coming to you from Lilly Human Health today. Elanco Animal Health is a part of Lilly, but the part of Lilly that I work in at this point in time is Lilly Human Health, Lilly Research Laboratories. And my background is in both clinical research for animals, developing drugs for animals and taking them through the FDA or USDA approval process. And then in the last 10 years, I've moved to the human side and have conducted clinical research with humans. And of course, as a veterinarian, that's a little bit unusual. I'm one of very few in our company that are non-physicians, although we have more and more clinical scientists coming in and doing working with our projects as we move them through human drug development. So I've worked on both sides. I've worked on the animal side as well as the human side. With that preface, I'll tell you that this timely valued medicines has become a very important aspect of what we keep in mind every day as we move forward. We're trying to constantly think about the patient, keep the patient at the center of our focus as we move forward. As we look down here, I want to draw your attention to the bottom of the slide. Let me use this pointer. And this bullet down here, or large arrow, features 5 of these TVM marks. And this is all underscoring this notion that we call next generation development. And this is how we believe we're going to start doing things a little differently in order to move things forward more effectively. Number 1 is a better disease understanding. Better understanding the actual diseases that we're working on, understanding the pathophysiology and the genomics or the molecular aspects of that disease. Tailoring of medicines is another aspect, making sure that we're developing the right drug for the right population of people or the right subpopulation of people. And this could also go for animals as well. The right therapeutic agents. Are we taking the right things forward? Are we being conscious of what we are essentially putting our money behind and developing to make sure that we're making the right choices of the medicines that we're taking forward? Robust phase 2 data, doing a really good job at proof of concept and dose determination to make sure that we're, again, moving the right things forward and not pushing things forward because it has a hint of efficacy, perhaps. And finally, differentiation in phase 3. How can this improve on what we already have available? How can it be better? So what we have in my area, one of the things that I am working on in our innovations efforts, over the last few months, I've taken on a new role, and we are looking more and more at doing translational and comparative medical research with naturally occurring diseases in animals. In other words, things like some of the things we've heard about today, cancer or muscular dystrophy or various infectious diseases or even areas such as pain. And we think that by using animals and the parallel diseases that those animals have, when it is parallel to the human disease counterpart, that we may be able to improve item 1 here, item 2 here, and item 3 here. Well, what's this all about? Why are we even doing anything different? Or why are we looking outside the box, so to speak? This is part of the problem, and this is Tufts University data from their Center for the Study of Drug Development, and this is from human research. The green line, it follows the number of new compound approvals. The area under the curve of the blue line shows the amount that's being spent in drug development for human disease in drug companies. And as you can see, the R&D expenditures is far exceeding the number of new compound approvals. This is untenable. We can't continue this and stay in business or have new drugs available for all the diseases that you all would like, we all would want us to treat. We also can look at this in a different way. This is clinical trial costs. And this is from a PharmaLot survey. It's a small article, but I think it's still useful to look at. These figures up here are dollar figures per patient when they divided out the cost of the study per patient enrolled on a human study. And you can see from 2008 to 2011, the costs of doing research are going up exponentially, and we don't see that changing. The last item I'd like to bring to your attention before we talk about some of the things that we're doing or the whys is where do drugs fail? This is phase 2 failures between 2008 and 2010, and this is phase 3 failures between 2007 and 2010. And these both appeared in Nature Reviews Drug Discovery in 2011. So phase 2, again, is the human area of research, is that area of proof of concept and dose determination, dose ranging. And over half of the phase 2 failures are due to efficacy. Over 2/3 of the phase 3 failures, or right at 2/3 of the phase 3 failures, are due to efficacy. And what this tells us is that we're not doing a very good job of predicting what is going to work when we take it forward into a human clinical trial. So that's one of the questions that we're trying to answer. How can we be better predictive, more predictive with all the tools that we potentially have available, and maybe even looking at some new tools? How can we be more effective and more predictive as we move forward into these very expensive human clinical trials? So we've got people over here on the right, and we know that they have diseases that we would like to treat and we would like to cure, whether it be Ebola, chronic pain, Alzheimer's, or a myriad of other diseases that people get that still do not have good answers. We've got silhouettes of dogs over here on the left side, and this could be other animals as well. But what we're looking for are parallel diseases that occur in humans that have counterparts in animals that we could potentially learn considerable amounts of information by doing more translational and comparative disease in the two animals. So we think by utilizing veterinary medical expertise as well as human medical expertise that we can move the entire one medicine concept forward. So by conducting one health-focused research using naturally occurring parallel disease states in animals, we think we have this opportunity in mind to increase the probability for human efficacy success, as well as open opportunities for animals, treatment of animal diseases, provide a cost, time, and savings to the overall development of drugs, increase the value of the early drug candidate package. In other words, when a company is making a decision or they're looking at selling it to another company, or perhaps even when NIH is making a decision on whether to take a novel molecule forward. To inform the overall safety data set, we gain more, as we heard today, we gain more information about the safety data set as it was illustrated in the Niemann-Pick disease cats. as we go into humans. We can provide also a potential alternate or parallel development strategy for veterinary medicine for use in animals. So it creates multiple opportunities, and it does nothing but help us gain additional information. So there's been a few examples in history of where this has happened, and probably the most mature area, as we've talked about earlier today, is in oncology. And so I borrowed this paper from Dog Models of Naturally Occurring Cancer back in 2011, and it shows the human development paradigm at the bottom and the animal health paradigm at the top. And essentially what we're wanting to do, what we're talking about, is translating back and forth. This has specific arrows going to different areas, but it could happen at any point along the progress of the development schema. And the information can inform each side, both the animal side as well as the human side, as things move forward. So the issues that I'd like to outline that we are encountering in the drug development world include a very high cost and a lengthy time in order to take a novel mechanism forward and find out whether it works or not in humans. It's a huge cost. The preclinical models that we have that are telling us whether we move forward or not, and frequently we are relying predominantly on those preclinical models. They're usually an induced physiological state in a purpose-bred laboratory animal, and they explain important aspects of pharmacology and pharmacodynamics, but they rarely represent the actual disease state itself. So we think we can introduce another aspect to the development schema that may help get us additional information to know which molecules we should be moving forward with. We're also encountering difficult decisions as to first-in-human studies with known concerns about the predictive value of the preceding data. So if we can introduce additional data to help us to stratify and decide on how we're going to move forward when we have multiple molecules, like in my company, we have multiple molecules to make a decision on moving forward. We can't develop all of them. We don't have the amount of money to do that. We also know that there's industry-wide concerns regarding how quickly we move into phase 3 studies. As a result, we tend to get excited. We say, gosh, we've got a fantastic new drug. It's going to treat XYZ disease, which is an epidemic, and it doesn't have any good solutions. And we tend to jump forward very, very quickly with a small amount of data. And sometimes we have to pull the reins in and remember to be more intentional and make sure that we get the important dosing data, dosing at the appropriate level. We get the important safety data before we drive straight into phase 3 studies. Finally, I've already covered with you the late stage issues of identification of efficacy and safety failures, which is really very unfortunate when we're running into those very late in development when millions and sometimes billions of dollars have been spent. So I'd like to give a few examples that I have found where drugs have first been developed in animals and later they've been translated to humans. But before I do that, I'll just reflect on the practice of veterinary medicine. Frequently, veterinarians use the armamentarium of drugs that are available for dogs or cats or some other species, and they don't have any other labeled choices available. So frequently, veterinarians are reaching for human drugs and using them off-label because there's no other choices available. As a result, we've learned a lot through the empirical evidence that has come through those uses on how things are translated from humans to animals. There's also, of course, much more data that is established in a more conventional research setting as well, and we know more and more what is translating to in both directions. So let's look at a few of the examples where it's gone from animals to humans. Ivermectin, the entire spectrum of ivermectin used across probably more species of animals than any, as many of you know, is an anthelmintic And it was after being on the market for some time for animals, it was became available through the WHO by a donation from Merck as Mectizan. And Mectizan was distributed to the areas where onchocerciasis is a huge problem and can cause a large percentage of the population to become blind. It was later approved for use in the United States as Stromectol, and it is labeled here in the United States for strongyloidiasis and sarcoptes in humans. We heard earlier today about samarium 153 from Dr. Carolyn Henry and the work at University of Missouri on this radioisotope. And this informed the progress forward to move into an approval for the human drug or radioisotope Quadramet. Another example is Palladia and Sutent, two cancer drugs that were developed essentially in concert, and the studies that were done informed each other as the progress in with these two molecules moved forward. These two molecules are very similar, but Sutent is labeled for humans, and my understanding is it's the standard of care for renal cell carcinoma, and Palladia is labeled for canine mastocytoma in dogs. The last one I've listed up here, I'll tell you a little bit more about in the next slide, but Optimmune and Restasis are cyclosporin ophthalmic drops for dry eye. There's another one I didn't list on here, and that is Lilly's spinosad, actually Elanco's spinosad, which is the active ingredient in Comfortis and in Trifexis. And it has been out-licensed by Elanco to another company for, and it is approved as a human lice treatment for kids. So the story behind Restasis and Optimmune for which is cyclosporin is an interesting one, and it goes way back to the 1980s, which seems a shorter and shorter time ago to me. This veterinarian, Dr. Renee Kaswan, actually holds the patent for the use of ocular use of cyclosporin drops for canine dry eye, or for dry eye in general. And she came out with this paper in August of 1989, and the legend goes that this came out and it wasn't taken very seriously by the human medical community. And consequently, it was 1995 when FDA approval came about for cyclosporin use for dry eye in dogs, and it was 8 years later that Restasis became available for humans. And if you haven't seen Restasis advertised on TV, you're probably not watching very much TV. It's quite amazing. So why, again, are we leveraging translational and comparative medicine in companion animals? We're proposing that we should do this. Naturally occurring diseases in animals that are parallel to human disease. I like to call them parallel diseases rather than models, because they're really people, especially at Lilly. I talk to people at Lilly and they hear model and they assume that I'm doing something to those animals to make it happen. We're talking about naturally occurring spontaneous diseases. So they're parallels. They're not exactly the same because it's not in the same species, but it's a parallel disease. We're talking about spontaneous occurrence, not induced. We're talking about physiology and pathophysiology that is often very similar, if not even identical. Exposure to environmental influences can be similar or the same as us, as we all live in the same homes with our pets. Active targets are often the same. We've proven that through our reverse translation and use of human drugs in animal health. Genetic causes may be homologous, as we heard earlier today about some of the analogous and homologous genetic causes. And this may particularly have important a role to play where we're talking about rare or orphan disease in humans. Rare disease or orphan disease status on the human side, for those of you that don't work on that side, is equivalent to the MUMS, or the minor use in minor species. It's a rough equivalent. It means it's a small enough population that it deserves special discussion in terms of how can you do the amount of research that you need to do, or what type of research needs to happen in order to get a drug approval. And finally, the treatment regimes in animals are often extrapolated from humans, as we've discussed. And we know that in the research we do with animals, we may be able to extrapolate more than just a yes or no, does this work, but even help allow that to help to establish treatment regimes and so forth. We also know that client-owned dogs and pets have owners that are motivated. These people are bringing their dogs and cats in to see a veterinarian because they are concerned about that animal, because they want something to happen. It's not just because they must do that. Obviously, they don't have to. These animals are usually well cared for. And for my human colleagues, I tell them, because sometimes they're not familiar with what I'm talking about, I tell them this is a very similar interface as you would think about a pediatrician dealing with a child. And then finally, I think we've heard this quite a bit, that veterinarians are underutilized as scientists, and we need to open more opportunities for veterinarians participating in research. We've seen this diagrammatic several times today. I guess it means it's a really good one, and we all like it. So other things that we can do, benefits that we can bring to the table, is that through translational and comparative medicine with naturally occurring diseases in pets, we have the ability to look into biomarker research. We have the ability to do imaging and repeat imaging, repeat other measures of different kinds across the course of the disease. We also have sophisticated inferences and observations from these animal owners, which we can't quite get in a laboratory setting. We don't have the same interaction with the animals to allow the same type of assessment. Of course, we still have the ability to have investigator assessments from a much more distant view, or a time point assessment of the animal, behavioral interactions, and repeat tissue collections as well. Finally, I'd like to just look at a few of the things that are coming up in the literature, just to reflect on all the changes that are happening right now that make this a really good time for us to talk about these opportunities more. This slide depicts two pieces of information that have been published recently. The top one is talking about OCD in dogs, which we've commonly called canine compulsive disorder. And they've actually identified a functional non-cloning variant in a canine model of OCD. We are seeing advances in genomics altogether that are helping us to identify how exactly where are the homologies or the analogous events and diseases going on in both animals and humans. We've already heard about the golden retriever muscular dystrophy dogs earlier today, and information continues to move forward on this dystrophin glycoprotein complex. And another one that I'd like to point out, I thought this was extremely interesting, as it came up a couple of years ago, I guess 3 years ago now. This PNPLA1 mutation causes autosomal recessive congenital ichthyosis in golden retriever dogs and in humans. And what they found was a new gene that was involved in human ichthyosis. It was identified subsequent to identification of the same gene in dogs. So as we think about these animals, and these breeds are like small colonies or small groups of people, similar to the Icelands and the other groups of humans that have been isolated to genetic diversification over time. So the research environments include clinical research, like we've mostly been talking about, but research colonies, like you've heard about earlier today, and finally, potentially with laboratory animals that have naturally occurring diseases such as obesity. So the time is right, better than any time. I think it's time for us to embrace this, to start to look at ways to pull both human medicine, medical research, and veterinary research together, and look at the opportunities for synergies. It's because of advancements such as what has happened here at NIH with the example that's been set with the comparative oncology program that Amy LeBlanc addressed earlier today, the biomedical developments in imaging and tissues, genomics, and the advancements that we have there. It's so much easier than it has been in the past to identify genomic causes of diseases. The One Health and One Medicine initiative that we're here for today, molecular profiling and the focus on personalized medicine, making sure that the medicine that is available to me is probably going to work on me, not just 80% of the people that were out there and treated with it. So we think that this is a great opportunity to help to move us forward more effectively in drug development research. Any questions at this point?
We're going to save the questions.
Okay, we're going to save questions.
Hi, everybody. My name is Kat Lutz, and I am the director for the Rare and Orphan Disease Center at the Jackson Laboratory, as well as the director for the mouse resources. Thank you. I'm here today representing industry in a strange way. I actually am not from industry. As many of you know, the Jackson Laboratory is a nonprofit organization. We're a research institute, and we're a provider of a number of different mouse resources to the scientific community. And our mission really is to do just that, is to provide the resources to the biomedical community to advance the research and hopefully find cures for some of the diseases that you're all working on. And I found this quite fitting, actually, because it's springtime almost in Maine, and there's lots of potholes out on the road. And so when you think about reproducibility and the road to drug development and those of using animal models, you think of a road that's pretty smooth and straight going ahead, but there are some potholes along the way. And so I just wanted to have more of a conversation about that than anything else today. And let you know a little bit again about my perspective and where I'm coming from. So, again, the Jackson Laboratory is a research institute, and we're also a distributor of mouse models. But the perspective that I'm giving you today is really from the role that we have in establishing phenotypically relevant platforms for preclinical development and drug development. So as many of you know, and you can imagine, a lot of biotech and pharmaceutical companies no longer have vivariums. They're looking at outsourcing, and they're looking at partnering with people who have the best knowledge about some of the mouse models that are out there. And they're wanting to test, some of their proof of concept, whether it's a repurposed drug or a new molecule in some of the mouse models. And so we've been trying to work with the our industry partners to help facilitate that. And we're also working with a number of foundations in rare and orphan diseases because, as you can imagine, for ALS and SMA and some of the very rare and ultra-rare diseases, it's not necessarily the case that those models are widely available or that people really know how to use them. And so we're also working and assisting in generating future models. And so and just as a background, I'll have to say I really have enjoyed all of the discussions today about the spontaneous models in dogs and cats. I have been a mouse geneticist my whole life, and I grew up and I cut my teeth on spontaneous models because long before we had genetic engineering, the only thing that we had to work on was spontaneous models. And so we looked for disease phenotypes in mice that looked like they had a clinical or a human component, and we went through and did the painstaking research that involved positional cloning and identifying the gene and looking at gene function. We've been very lucky in mouse genetics to be able to take advantage of a lot of the genetic engineering techniques, and I'm going to talk a little bit more about some of the newer techniques and how we can really harness them to our advantage. But before I do that, I just want to talk about the mouse as a model, in general, because I think, in some ways the mouse gets a bad rap. This is an inbred mouse. there's no way that it is going to phenotypically represent the entire population. It's inbred by nature so that we can use the power of the fact that, genomically and genetically, it's very, very stable. But because of that, it lacks the heterogeneity, the genetic heterogeneity that we see in a population. So it's a little unrealistic to expect a mouse model to fully recapitulate any aspect or all of the aspects of a human disease. But I think if we gauge our expectations and understand what the mouse model is good for, and then also realistically what it's not good for, we can use the mouse model, to an advantage in such a way that it can inform clinical trials. And so, because I have 2 teenagers, I've become very conversant in text messaging lately. And so I can say truth, because that's what my daughter says all the time. There are 65 million years, of evolution between mice and humans. And so we need to get over that. Get over it, GOI, oh my God, right? And really understand, what the limitations are of the mouse models, because, so many times when I work with industry partners, they're looking for example, in spinal muscular atrophy, they're looking at a mouse model and they want to use survival as their preclinical outcome measure. And the fact of the matter is that those mice don't die of spinal muscular atrophy. They die of cardiac hypertrophy, which we don't see in humans. But there are other biomarkers that are quite valuable. So you have to know the model in front of you, what it can tell you, and most importantly, don't fool yourself into thinking that the mouse model is telling you something it's not. So we need to be realistic. So 2 of the things, there's all kinds of topics we can talk about mouse models, their goodness, what they're good for, what they're not good for, and various things. But one of the things I want to really focus on today is how we can use the resources we have better. What have been the potholes that we've seen in the roads, and why can't we necessarily use these models to the best of our advantage? And so I think the first part of it, really deals with accessibility. And so I told you I, sort of cut my teeth on genetics as a researcher. And when I started working more in the resource aspect of science, I started as working in the mouse repository, working with operations, getting people to donate their mouse models to public repositories so that we could stabilize the genetic background, have a high health standard, associated with these mouse models, and really not confuse what is, a somewhat confusing base of scientific literature out there. And so, really, when you think about accessibility, researchers who engineer their mice are often limited by their ability to provide them to the scientific community, especially under these high health statuses. There's nothing worse than parvovirus in your colony, to make you, really have some scientific results that aren't necessarily real. And then it just really expedites the process. People can call, they can order the model, they come from high health status, and then there's just, widespread accessibility. And certainly the Mouse Mutant Regional Resource Center, for which Jax and my partners at UNC, UC Davis, and Missouri are a part of, have played a critical role in making sure that the mouse resources that are developed with NIH money are actually getting to the people who really need to use them. And so that's been great. But still, I think some people find it hard to share their models. And this is sort of a dichotomy that we've set up, within academia itself, because when you make a mouse model as an academic research, that's your ticket, right? That's your ticket to new grants, that's your ticket to papers. And when you're coming from a small lab and you're being told, if you want a tenure track position and if you want to keep your job, you need to have bookend publications in Nature and Science. Well, you're not necessarily going to get those bookend publications in Nature and Science if you make a model and you give it away, right? So people have a tendency to hold on to these models much longer than they should. And the fact of the matter is it could be years before these models make it into a public repository. And then the people who are making them sometimes are using cost-effective measures and efficiencies, like they're making their models in CD1 and mixed genetic backgrounds. So what they have today isn't what they have tomorrow. We've had limitations on ES cell lines that give us, mixtures of B6 and 129. So now we start to see differences in genetic background. We see differences in genetic drift. And guess what? The model that you made 3 years ago is phenotypically very different than the one you have today. And we see it all the time, right? So no surprise that there's problems with reproducibility in the scientific literature. So the other additional pain point that I think, especially that I've seen in working with biotech and industry partners at this point, is the pain of tech transfer in using the mouse models. And so as some of you are aware, especially at NIH, the Bayh-Dole Act was a congressional act in the 80s that gave ownership of inventions to federal funding. It was fantastic in stirring up competition and capitalism and really getting research institutions invested in ways that were just a recipient of a grant that went away. But the problem with that, so basically the research institutions who made these mouse models now had the ability to patent and license them, which is good for them because now the research institutions have a way of profiting off of the invention and their investment. And of course, NIH has a sharing policy that says, please deposit your mouse models. And so they deposit it into the Jackson Laboratory or UC Davis or one of the other MMRC units. And all is good in the world if you're working in academia. If you're not working in academia, it becomes very, very difficult to use these models. And so when you have pharma and biotech up here in the corner who say, hey, I've got this great drug that's going to treat this disease, whether it's diabetes, obesity, or rare and orphan disease, and they're going to say, hey, MMRC folks, hand me that mouse model. And there's going to be a big barrier there. And that's going to be the research institution's tech transfer office, who's basically going to say, we'll negotiate that license with you. This is a nail in the coffin for biotech, especially when they're working with very limited timelines and very small budgets. And they inevitably say to us, when they're wanting to work with an ALS model or an SMA model, the first thing they say to us is, show me all the models that you have that don't have a license. That's not very many. And so what they end up doing is sometimes using mouse models that are not the best models, but the ones that can give them an answer the quickest. And so the problem that I see from my perspective of dealing with tech transfer offices is that pharma and biotech are constantly going back and forth with the tech transfer offices from these research institutions. And maybe you can say that that's just, inconvenient, but for some cases, it's a real deal killer. And when you think about the new technologies and the number of mouse models that are going to be generated over the course of the next 5 years, that's going to be unsustainable. We can't do that anymore. If we can't facilitate this process, biotech and pharma use of animal models really becomes prohibitive. So what I hope I've done is try to convince you that, between the academic goals for people who want tenure-track positions and IP and patented, it's really not in the best interest of the patients because we need to get the resources into the hands of the biomedical community who are going to use these to find treatments for diseases. And while I respect all the faculty members, aspirations to have a tenure-track position, and I also respect the research institutions' rights to patent, these things have to happen more quickly and they have to be better facilitated. So I think that kind of leads me to what I'm really excited about in the field right now is the disruptive technologies that we have, and those are whole genome sequencing and gene editing technologies. And so I'm not going to spend a lot of time on CRISPR-Cas9 because unless you live in a cave, you pretty much know exactly what these technologies are. But what it really means to us, especially in the mouse resource area, are better ways of making precise genomic alterations. And this isn't just, taking a neomycin cassette and blowing a hole in the protein and saying what happens, right? It's taking very specific mutations that exist in patients and having the ability in a very short period of time to take a zygote, inject it with a guide and an oligo, insert it into a blastocyst, come out with animals that for a certain percentage of the cases, carry that mutation. And there he is right there, doing an in vitro fertilization And in less than 2 breeding generations, you've got all the animals and all the cohorts of animals that you need to test and characterize your phenotypes in your drugs. And so this is incredibly powerful when you think of where we were before with traditional gene targeting, where it took, a few years, quite frankly, in order to accomplish this. And because we had a talk on muscular dystrophy, I just wanted to say we've used these genome editing technologies to put mutations that were once in the C57 black 6 mouse, which admittedly is true. It's a very mild phenotype. It can be not informative at times. And we put it in the DBA2J, and we have a spectacular phenotype now associated with increased fibrosis and inflammation. And so this is a question of optimization. And so what's really cool about the genome editing technologies is you can put these into any genetic background that you want. And here is where we've put them in all of the progenitor strains of the collaborative cross. And so if you're not a geneticist, that may not mean anything to you. But it's really powerful now to be able to do genetic engineering and genetic editing in strains other than C57 black 6 and 129. It's incredibly powerful. So, the advantages are, that it's inexpensive and it's fast and you can inject straight into zygotes. There's no ESLs. There's no breeding away of selection cassettes. There's unlimited use of genetic backgrounds, and you can make therapeutic platform-specific mutations. And so if you have, a point mutation and you want to, or a stop codon, and you have a drug platform that's for read-through drugs, you can make that precise model, that precise human mutation in a mouse model. You can test exon skipping technologies, antisense oligonucleotides. All that becomes possible now in ways that we just didn't have before, because the technology, while it was there, it was inefficient and imperfect. So this is where I think, as a community, we need to come together as one health and one organization and find out and really take advantage of how these disruptive technologies change your business model. How is this going to change how we work every day with one another, both as from the research perspective, from repositories, as well as from industry? And so this is one of my favorite slides ever. And this is just a graph of Kodak's stock. And Kodak, actually, as you know, was quite an industry heavy in the picture and film industry in the early 1990s. But what Kodak failed to do is embrace the disruptive technology of digital photography. They held on to their handheld pictures and said, it's not going to change. Everybody's going to want to take a paper photograph and hold it. And guess what? They lost. That's not what happened. Digital photography, took over, and Kodak stock went down the drain, and they dissolved almost primarily as a company. So I think it's really important to really understand how these disruptive technologies are going to change how we do business. And so when I think about that, I get a little scared. I think, these are the best of times, and as you can tell, I'm really excited about it, but then I think it's the worst of times, right? Because I think the implications for this technology are great, and that, individual investigators won't be able to sequester these mouse models in their lab anymore. Game over, guys. I can just remake that model, and I can remake it in 10 weeks, right? So for those of you who are holding on to them for 5 or 6 years and won't give them to the public so that they can actually use these for drug discovery, it's not going to happen anymore. We're just going to remake them, right? And we have the technology to do it. And the barrier of time and expense no longer exists, right? So that's amazing. And we can use these for precision modeling for specific human mutations to really address, what happens with these platform drugs. And again, talk about genetic modifiers. In most of the cases, we're not going to be targeting the gene itself as a therapy. We're going to be targeting modifiers. And what better way to understand what the modifiers are is to start moving around in different genetic backgrounds and get to those modifiers, because those genetic modifiers are your next drug target. So how cool is that? It's really cool. The negatives, I think, that are going to come with CRISPR-Cas9 technology, and this is where we have to strategize as a community and really come together, is that there is going to be a tidal wave of mouse models that are going to be made into the scientific community. I can't tell you how easy this technology is to use. We can make knockouts. Knock-ins are a little harder, and you moving big pieces of DNA around are not the easiest to do, but these are the infancies of this technology. And so it's just a matter of time before we figure it out. And for the most part, we already have. So you don't want to get to the point where you have disposable mouse models, right? You don't want everybody saying, oh, I need a new mouse model for MDX, for muscular dystrophy, and I'm just going to remake it in my lab, and when I'm done with it, I'm going to throw it away. Because now we're going to have problems with reproducibility like we've never seen before. So there's a caution that comes with this technology that I think that we need to be aware of. And I also think the negative part of this is that you no longer have to do crossbreeding to introduce new mutations onto other backgrounds. And so if I have an Alzheimer's model, that's okay for making Alzheimer's, but now I've identified a new gene in a GWAS study that's going to make that better, and I make it, suddenly you're going to have what I refer to as oligo-licensing, right? So now everybody's going to carry a license on that, depending on where that was made. And so suddenly what you think is a really reasonable license for your mutation, when combined with other mutations, are going to be a license that's going to cost $100,000 for a biotech company to look at it. And guess what? They're not going to do it. So I think in a lot of ways, the challenge is, can technological innovation lead to process innovation? And how do we, as the thought leaders in this field, make sure that we're going down the path with the least amount of potholes and no sinkholes? And I think that really isn't the mouse in the room, it's the elephant in the room. And I use ALS as an example. I'm a neurobiologist by training, and so most of the work that I do is in neurodegenerative diseases. And I can tell you that for years and years, the only mouse model that we had for ALS is SOD1. And for everybody who uses the SOD1 mouse model as a perfect example as to why mouse models don't really translate into good clinics, should have their degree rolled up and beaten with it. Because the fact of the matter is the SOD1 mouse model was the only mouse model we have, and it represents almost 2% of the entire population. So to take a drug that seemingly worked in SOD1 and put it in a clinical trial where you don't even know if those patients have SOD1 is not reasonable. It's risky. And so, but that's a problem with the genetics. It's a problem with the clinical trials. It's a problem with the patients, who die so quickly of this disease. It's not really indicative of a bad mouse model. It's just an unfortunate set of circumstances. And so what's really interesting, I think, is that with genome sequencing over the last few years, we've identified all of these different causative genes now for ALS. And I work very closely with the ALS Association, and I was really struck when TDP43 came out on the rise, and they said, Kat, make sure that you're working with all of the ALS investigators to get the TDP43 models. And guess what? They all made them the same way. They all made them on mixed genetic backgrounds. By the time we got to them, we couldn't figure out anything. And it took us almost 3 years to realize that what was first identified as a motor neuron degeneration in these mouse models was not motor neuron degeneration. It was a disease of neurodegeneration, but it was in the myenteric plexus. It had nothing to do with motor neuron disease, and that was a factor of having moved that mouse model on different genetic backgrounds. So at the end of the day, we still don't have a very good mouse model that represents TDP43, but we spend an awful lot of time and money, trying to do that. And so I always refer to this now with all of these different genes that have been identified as the mouse bucket challenge. And I think that if you look at this, you can insert any disease in here and find out that it would be really interesting. And so the idea is to not duplicate and not to duplicate efforts and not to waste resources. But what if we broke the mold? And what if we took the box and we threw it away and we said, you know what, we're going to make models now through a third party. We're going to remove aspects of oligogenic licensing, and we're going to make these mouse models available. And we're going to make them available as soon as we make them. And so we use a model of crowdsourcing to characterize the models. And the researchers actually spend their time characterizing and studying the mouse, not making it. And so, again, I think this is a new business model. I think it's one that we need to seriously consider. I think it starts with, a lot of different steps, pivots, and iterations of our current business model and how we work things. And we move more towards the globalization of these resources in a way that's economically feasible. And hopefully it's about as liberating in that case as the Berlin Wall coming down. And I think just like with the drug, I think with mouse models, you make it, you distribute it quickly, and you kill the model quickly. If it's not any good, let's find out quickly, and let's stop, confusing the scientific literature with a bunch of information that's not correct, and let's move on. You crowdsource the characterization, you standardize the tech transfer, you ensure the reproducibility, and most importantly, you serve the patients. And so just to say that we have done this for C9orf72, this was a collaboration I did with Bob Balow. We found that the mouse model recapitulates a lot of the pathological phenotypes that are seen in humans. And most importantly, this mouse model isn't even published yet, but as soon as it does become published, this is my collaborator, Bob Balow, those mouse models will be available to anybody who wants to have them with very limited licensing and very reasonable tech transfer. So I do think it's the best of times. I think there's a lot of opportunity, and I think that we really should embrace it and take advantage of it. And with that, I will leave you with this beautiful picture of the Jackson Laboratory. That's my office in Bar Harbor, Maine. And for anybody who wants to come and visit, I encourage you to. Thank you so much.
We probably can have only a quick question for any one of our last two speakers. Yes, we have two questions here.
Yeah, I have just, you actually touched on something I've been wondering about. Galen Edwards from the University of Georgia, wondering about all day, and that's reproducibility. Reproducibility has made nature and science many times in the last 2 or 3 years, and it's becoming a huge problem for animal research, in that PETA, PCRM, HSUS are picking it up. And unlike most of the people in this room, they are on Capitol Hill, and they're lobbying really hard to cut funding to science. So, how do we address this? And I'll open it up to everybody in the room. It's a big problem, and NIH has an initiative now to address it. So.
I mean, I think it's a really great question. It's something that my collaborators at the MMRC, Kent Lloyd and Craig Franklin and I are talking about constantly, and how can we better serve the community? And I think that really, in this case with C9orf72, and this is what we're doing now with a number of ALS genes that have been recently identified, it benefits the ALS Association and the patient community not to have their mouse models sequestered or licensed in a way that's not reasonable. But most importantly, when we have the mouse models, we're doing second site validation, as well at the same time. But really this aspect of, because nobody knows everything, and you're going to look under the street lamp. You're going to look at that mouse model for what you know. But as soon as you make it available and you crowdsource it, everybody's going to know everything that needs to be known about that mouse model really quickly. And it's not going to take 5 years. It's going to take 1 year.
Yeah, I agree. Sharing it among scientists is great, and also the other models in clinical cases are great. I've heard several comments today though about the low numbers of animals in studies, and when you go back, if you look at the studies that are criticizing reproducibility, the low-powered studies that lead to clinical trials that fail in efficacy. It just happens over and over
again. Right. And again, I think if you work with a CRO, or you're making sure that your studies are well powered and blinded and randomized, and those are all things that with a little education, I think that we can definitely get there. But again, the accessibility, I can't tell you how many biotech companies come to me and say, show me a list of your models that don't have licensing associated with it, because they've got X amount of time, to get this proof of concept to their management. And if they've got to fool around for 5 months with the tech transfer office, it's a deal killer for them. And so the problem with then is that you're doing your preclinical studies on a less than an ideal model, garbage in, garbage out, right?
I actually had a question for Carol. As you probably are aware, your company is in the minority in terms of using the companion animals for your studies. And I wondered if you would comment about that regarding perceived risks of placing human destined drugs in companion animals for clinical trials.
I do realize we're in the minority. I do think we'll see some change with that over time, and it still is a new thing for Lilly by any means. This is really just started over about the last year, And I've come into this role just in the last 9 months, and so we're really making things happen and move forward now that there's somebody able to dedicate some focus to it. Perceived risks, you know, I think that the risks are greatest regarding the perception that the diseases are not similar enough. And it's this lack of realization that we really do see cancer in dogs, and we do see pain in dogs and cats and across all the different species. In terms of with specific molecules going forward, I think that there is sometimes some concern. There's been voiced concern in the past, while you're putting the molecule at risk, you may see an adverse event in animals that we don't see in humans. I think that that's perhaps more valid if you're later in drug development. If you are in phase 3 with a human molecule and you know what you're going to see, you've already dosed hundreds and maybe thousands of patients, and so you know what the adverse event profile looks like. And the concern then is, oh, geez, but if we go into dogs, one of them might have a seizure, because we know they're little seizure machines, and every once in a while they have seizures that we can't explain, or convulsions we can't explain. Then I can see there's a little bit of concern that possibly could be legitimate. But if we're planning ahead and we're putting this into the whole construct of how we're going to do drug development, then I think we can mitigate those perceived risks. And I think those risks are very, very low, even if you're talking about late stage.
And where along the continuum of drug development do you see the dog models going in? Is it like the drawing where they go in parallel in phase 1?
I think
and inform phase 2.
I think there's several places that we could see them work, or these parallel animal diseases work. The easiest place to fit them in that addresses this perceived risk issue is before you even go into humans. So put it in the overall plan, the overall schema for the drug's development. And if you have questions that can't be answered by your models, then, and they can potentially be answered with an efficacy study in spontaneously occurring disease in companion animals, then that's the place to put it in. Potentially later on, as questions come up during drug development, it's possible we can address some things in animals, in spontaneously occurring disease in animals, perhaps on dosing schema and dose reductions or split dosing. We might be able to do that more quickly in animals while human studies continue. But again, that gets back to the risk issue or perceived risk issue you mentioned.